So we're going to look at the first part of this new unit with cell theory and microscopes. Cell theory starts with the discovery of the cell. In 1665, Robert Hooke was using an early compound microscope to look at thin pieces of cork. Cork is the bark of a tree. Same thing that you find in things like wine bottles. He took a thin piece of cork, put it under one of these early microscopes, and what he found were tiny little empty chambers. These little empty chambers reminded him of the little cubicles that monks used to live in. These little cubicles were called cells. So he called these little compartments he saw in the cork cells. Cells will end up being the basic units of life. Here we see a picture of Hooke's microscope, very primitive technology compared to what we'll be using in the lab. And we see a drawing that Hooke made of the cork cells that he observed. Now, it's important to note that these cork cells that he observed were actually dead cells. There was nothing inside of them. They were just the empty cell walls of once living things. The first person to actually observe living cells would be Antoine van Leeuwenhoek. He had his own little microscope, this tiny little thing you see him holding up to his eye. And in that tiny little microscope, he was able to see the first living things in 1677. His microscope had only a 200 times magnification. The microscopes that we use in the lab have twice that much power. The next person to contribute to cell theory is Matthias Schleiden. Using more modern microscopes, because here nearly 200 years have gone by, so obviously the technology will have improved in that time. Using these newer, more powerful microscopes, looking at various plant samples, he determined that all plants are made up of these cells. Again in 1838. A year later, in 1839, Theodor Schwann did the same thing with animal cells. After looking at various animal cells under microscope, or animal tissues under microscopes, he determined that all animals were also made up of cells. So basically did the same thing for animals that Schleiden did for plants. A few years down the road, 1855, Rudolf Virchow would be the last person to contribute to our ideas that lead up to cell theory. What he concluded from his observations that new cells were created only from the division of existing cells. So simply put, when you eat, you're not eating to get new cells. You're eating to get the compounds that you need so that the cells that you already have can divide and make new cells. Again, all these ideas are combined together into cell theory. Cell theory is a combination of all the ideas that everyone from Hooke through Virchow came up with in a single unified idea or theory. Cell theory says three things. All living things are composed of cells. Cells are the basic units and structure and function of living things. They are what make living things tick. And new cells are produced from existing cells. It's important to understand that these things that we're talking about are far smaller than what can be seen with the human eye. The human eye can see down to about 100 micrometers, which would take you down to the width of a hair. Cells are down around the size of 10 micrometers, so 10 times smaller than what we can see with the naked eye. The development of the microscope was essential for the development of cell theory. Cells would never have been identified. Cells would never have been attributed to plants and animals and established as the fundamental things and units that you find in all living things without the invention of the microscope. The original microscopes that these scientists were using were light microscopes, and that's what we'll be using in the lab. We will use a compound light microscope, compound meaning there's more than one lens grouping in it. As what we call an objective lens and an eyepiece lens that combine together to magnify the object that we're looking at. Compound light microscopes can see a lot of things, but most of those things must be stained before you can look at them under a microscope. That's why everything you look at under a microscope has kind of a pinkish tinge to it. That's the stain that increases the contrast and allows us to see what we're looking at. These are some pictures of what you would expect to see under a compound light microscope. Compound light microscopes go up to about 1,000 times the magnification. The ones that we're using in the lab will go up to 400 times magnification. And that does allow us to see the cells themselves in the larger organelles like the nucleus. Those are what the dark red spots are in that bottom picture. If we want to see anything smaller, like the organelles that we find in cells, we need electron microscopes. Instead of seeing things with light, they see things with a beam of electrons. Electron microscopes reveal details 1,000 times smaller than those you can see in a visible light microscope. So we can see organelles, the things that make up the cells. One of the drawbacks, though, to the electron microscope is the things that you view under them must be preserved. They must be dead. The special techniques that are required, the shaving them into thin sections, the coating them with metals that you see in some electron microscopes will essentially kill whatever you're trying to see under them. So no living things. There are transmission electron microscopes, TEMS for short. Transmission electron microscopes require the object being observed to be cut into very thin slices. Obviously, if you're taking something living and cutting it into very thin slices, it won't be alive when you look at it in the microscope. Like I said, you only see preserved specimens under one of these TEMS. But it does allow us to see greater detail. We can see the chromosomes in the nucleus here. We can start to see some of the organelles in the cell, a lot finer detail than what you can see with a light microscope. Scanning electron microscopes allow us to produce three-dimensional images, SEMS for short. The object is coated with a thin layer of metal, and then a beam of electrons is used to scan over it and produce a three-dimensional image. So rather than just seeing a thin slice of what you're trying to look at, you see the whole object in 3 dimensions. It gives us a great detail on what the object really looks like. There is a third type called the tunneling electron microscope. These are the most powerful of all the electron microscopes. These are the ones that we've used to see actual molecules and atoms. So they're more into the chemistry and physics than they are in the biology. So there you have it. In addition to this, we went through a reading assignment on microscopes to start learning the parts and pieces of them so that we can use them in the lab. I will attach that with this video.